Okay. All right. So here's where we left off with a little bit more on this heritability calculation. So one of the challenges of this is that we've set up a really kind of arbitrary system. It's the best, probably one of the best things that we have, but it is still not perfect. A major assumption is that the environment is the same between our monozygotic twins versus our dizygotic twins, and that cancels out. If there is a greater degree of environment sharing, a difference between monozygotic twins and dizygotic twins, it will throw our numbers off. You already saw this. Here are dizygotic twins. These two in particular are known. These are dizygotic twins. They don't necessarily look the same. Their parents tend not to dress them the same. Now, these are two different sexes, and so also true. And so if you were to then say, what's the heritability of clothing choice? This would give you an artifact. It would suggest that there's a very strong genetic contribution of what you wear every day, if you are monozygotic twin versus dizygotic twin. Clearly, that's not the case. It's not genetic. At least we're assuming it's not strongly genetic. But there's an environmental difference, and so it will throw this off. So a critical thing here is to realize that these are estimates for heritability. They are not absolute numbers. They're a best guess for these things. So just a couple of general principles. Recurrence risk is higher if the disease is more severe in the proband. So what are we saying there? Essentially what we're saying there is more severe expression of the particular disorder suggests higher liability. So a child expresses a very severe disease, we would put them up here. Common question that the parents are going to ask is, what's the likelihood that this is going to happen again? If we place the child up here, another thing that we would do is we would probably estimate that the parent, one or the other or possibly both, is also very high on the liability threshold. So we might put the parents way up here. Whereas if we have a child who exhibits a more mild form, the parents are likely to be a little bit lower. We're guessing, but we're trying to use an educated guess. So a more severe phenotype, more likely that it's going to reoccur. Again, to go back to the breast cancer. You can have unilateral versus bilateral breast cancer. In other words, are both breasts affected or is just one? Having bilateral breast cancer is a more severe clinical presentation. That would suggest greater liability, greater risk of recurrence if it's bilateral versus unilateral. Another one. Recurrence risk is higher if the proband is of the less commonly affected sex. We have already talked about this already. So for multiple sclerosis, it takes more for a male to exhibit MS than it does for a female. Therefore, if we saw a male who was affected with MS and then he had an offspring, that would be a greater risk of reoccurrence than if it was a female who had MS and had an offspring. And then the greatest risk of recurrence of all of the different possibilities would be a male with MS having a female offspring. And hopefully that makes sense. If it doesn't make sense, definitely come and talk to me or send me an email. So less commonly affected sex, higher on the liability threshold. Pyloric stenosis, it would be a female having pyloric stenosis and then having a male offspring. Recurrence risk decreases rapidly with increases in the degree of relatedness. So data for cleft lip, clubfoot, congenital hip dislocation. Prevalence in the general population, you can look up here, the first degree, second degree, third degree. Are you familiar with these terms, first, second, third degree, or no? No, okay. First degree essentially means the nuclear family. This would be offspring plus parents. So parents plus offspring, first degree. Second degree goes one generation beyond that. So that would be grandparents, for example. Third degree would be one generation further than that. So great-grandparents would be an example of a third degree relative. So if you look at cleft lip, in the general population, 0.001% of the population affected. First degree, so we have an individual as cleft lip and then a first degree relative, and we're asking the first degree relative, and that would be 0.007, or sorry, 0.04. Second degree, 0.007, third degree, 0.003. All of this is determined epidemiologically, just looking at the data. So I mentioned earlier this idea of, my great-grandfather had multiple sclerosis. What's the likelihood that my offspring could have multiple sclerosis? It's going to drop a lot, and it's going to drop pretty rapidly as well. And it's different for each disease. So in this particular case, it comes pretty close to the general population by the time you get to the third degree. So largely, when we're talking about multifactorial disorders, what we're looking at is first-degree relatives in terms of being affected. The idea here is that there's a very unique combination of genetic liability and environmental liability that has to come together to produce the disease. And the likelihood of this happening again is much less likely as you get further and further away from the individual who had it, because you're introducing new genes, new different polymorphisms, different alleles, et cetera, that then probably dilute the effects of those genes. So cleft lip and palate, recurrence risk decreases 13-fold from first degree to third degree relatives. I will do this very quickly because I don't want to dwell on this a whole lot, and I'm not going to require you to know these data. But this is kind of interesting in terms of what we're kind of doing with these disorders. So genetics of multiple sclerosis. There are multiple different subtypes of multiple sclerosis. First one is MS type 1. It is associated with variation on HLA genes. And oftentimes I don't require students to know genetic locations unless it's like on the X chromosome. But I think you should know where the HLA genes reside. And they're on chromosome 6, 6p21. And that'll make sense when we do cytogenetics a little bit later on. But chromosome 6 is where the HLA genes. MS, and there's 4 different particular HLA alleles on chromosome 6 that are associated with MS1. MS2, interleukin-2 receptor alpha gene, chromosome 10. MS3, IL-7 receptor, and a gene called C7 that's involved in complement. MS4 is a KIF1B gene, and I forget what that one does. And then MS5, tumor necrosis factor receptor SF1A gene. And again, you don't have to know any of these genes. But just there are multiple subtypes of MS, and there have been multiple genes that have been associated with these subtypes. So one of the things that people are trying to do is try to identify these many, many genes that are contributing. So the International Multiple Sclerosis Genetics Consortium and the Wellcome Trust Case Control Consortium, this is a hyperlink, by the way, and you can click on it. And you can see the results of this. They did what is called genome-wide association studies, GWAS. And genome-wide association studies, I will be talking about in more detail in one of the later lectures today, actually. But essentially, by doing these genome-wide association studies, they found 49 distinct genomic sites that contribute susceptibility to MS. So we talked about with height, there's about 400 or so. For MS, there's at least 49 different genetic sites that are contributing to susceptibility. What combinations are required is unknown at this point in time. So we're identifying genes that are contributing, but we don't have the whole repertoire at all at this point in time. This is kind of what a genome-wide association study looks like. And I'll show you another example of this a little bit later on. These are all different genes that have been identified in these genome-wide association studies, and then how strongly they were linked to these. So the way that these work, and I'll introduce this and we'll come back to it a little bit later on as well, is that we look at SNPs, single nucleotide polymorphisms. And hundreds of thousands of these have been mapped to the various chromosomes in humans. And the idea here is to segregate two large populations. And essentially we have those who have the disorder. So in this particular case, they have MS They have been diagnosed with MS, and then we have controls who do not have MS. And they have been screened to be sure that they don't. And then we are simply saying for a particular single nucleotide polymorphism, a variation in the population, let's compare these two populations and see if there's a difference. And then we can do lots of mathematical analysis. And for this particular one, what we find is that this is p equals 1/10 to the minus 12. So we see a difference. We're saying that this is the typical homozygote who doesn't carry the mutation. This is a heterozygote who is heterozygous for the mutation, and then a homozygote for that particular mutation. Is there a difference in the populations? So we see that in the cases, it's enriched. And this value is what's the likelihood that this enrichment was just by chance. And that's 1/10 to the minus 12. So a very small likelihood that that was by chance, that variation. And then here's another one that did also show a correlation 1/10 to the minus 8, where the cases are enriched for that particular variant versus the controls. It is a fishing expedition, just screening SNPs, polymorphisms, another after another and after another, looking for something that's going to match up. The vast majority of the SNPs that are analyzed are down here, where the log of the odds, which is a log score, is very, very low, too low to be significant. But then a few of them pop up. And so these two are particularly high, and then we can take those and we can say, what are the genes that are nearby? What are the genes that are close? So here's what was done with MS. And so each of these was rather high in terms of this enrichment in the disease population relative to the control population. We can isolate those genes, we can characterize them and try to figure out what do they do? How are they involved? And it's possible that they're not involved. Maybe there's another gene that's close by that's actually involved. So these may be involved, but we have to do a whole lot more work. So these genome-wide association studies have created lots and lots and lots of linkage to different genes, and now it's the process of trying to figure out what that means. Okay, that wraps that one up. Before I move on to the next one, any question on multifactorial in here? Probably not the slide I want. Okay. Next thing we want to talk about is epigenetics. And epigenetics certainly kind of plays into multifactorial inheritance quite a bit as well. So again, I have objectives for you. Definitely look through those, make sure that you're able to answer all of these objectives in full detail. If not, you can certainly send me an email. Once I'm gone, you can definitely email me as well. That happens all the time. So no problem. So what do we mean when we say epigenetics? How many people have heard of epigenetics before? Probably quite a few. Okay. So literally, it means above the genetics. And what we mean by that is that epigenetics is not a change in the simple base by base by base sequence. A, T, C, G, A, G, C, A, T, G. There's no change in that. But it is a change in function for a particular region of the material. Had a lot of definitions. The one that I like best is this one. It was from a Cold Spring Harbor meeting about 10 years ago. Epigenetic trait is a stably heritable phenotype resulting from changes in a chromosome without alterations in the DNA sequence. So the DNA is somehow altered, changes in the chromosome, but not changes in the DNA sequence itself is an epigenetic phenomenon. So alterations in DNA sequence, those are the mutations. This does not involve mutations, but it does contribute in terms of presentations of diseases and presentations of phenotypes. If you look at the number of publications in the last 20 years or so, huge increase in terms of epigenetic publications starting in about the year 2000 or so. So in the last roughly 20 years, we've really seen an increase in terms of epigenetics research. We have understood, or I don't want to say understood, we have observed the effects of epigenetics for a long time. The Lion hypothesis was from 1961. And this suggested that there's a difference in females between the two X chromosomes. And so when you had X-linked inheritance, we probably talked about this a little bit, that all females, in terms of their somatic cells, have one of the two X chromosomes preferentially inactivated. This is the Lion hypothesis, renamed the Lion law in 2011, because essentially it was found to be absolutely true, largely true. The inactive X chromosome is heavily epigenetically modified. So epigenetics is responsible for the shutting off of the inactive, mostly inactive X. This is an example. Here is the inactive X, and what we're looking for is methylation on that chromosome. And that chromosome is highly methylated, and that corresponds with shutting off of the sequences. Is the DNA changed? Are the sequences changed? They are not. But there is an epigenetic modification. Another familiar epigenetic case is on chromosome 15. Chromosome 15 imprinting is responsible for these two distinct syndromes, Prader-Willi syndrome versus Angelman syndrome. Actually, just before I left, there was an article in the newspaper that was talking about a 2-year-old who had been recently diagnosed with Prader-Willi syndrome. And I always hate when they do genetic stories in the newspaper because they never talk about the genetics at all of these things. And I'm always looking for the genetics and I don't see it. The worst one is, and I'm sure you've seen this before, new gene discovered for X, Y, or Z. What's the name of the gene? They don't even say in the article. You just go, oh, come on, at least say the name of the gene. But Prader-Willi syndrome, Angelman syndrome, two distinct disorders. They are linked together. They are both on chromosome 15, and they're both influenced by chromosome 15 imprinting. So how does this work? You're very likely to get a question one way or another on Prader-Willi Angelman syndrome on the boards as well. Everybody perks up when you mention boards, so it's always a good thing to throw out. What's happening? Essentially, in most cases of Prader-Willi or Angelman, we're dealing with a deletion on chromosome 15. It is a spontaneous deletion that occurred in the germ cell of one of these two parents. So neither parent had Prader-Willi or Angelman, but then they have an offspring who does. So in this particular pedigree, what we're seeing is the deletion is coming from the paternal germline. The chromosome 15 that was donated by the father has a deletion. The deletion takes out a particular part of chromosome 15. It includes a Prader-Willi syndrome region and an Angelman syndrome gene. There's one gene that's responsible for Angelman syndrome. There are several genes that are responsible for Prader-Willi. That's why we call that Prader-Willi region, because it deletes several genes. So if this deletion is passed on by the father, and the mother passes on a normal chromosome 15, the child will have Prader-Willi syndrome. Here is the exact opposite case. Here, the mother is passing on the same chromosomal deletion, not exactly the same, but essentially deleting the same region. We're saying these are unrelated pedigrees. So deletion here has also eliminated the Prader-Willi syndrome region, the Angelman syndrome gene, but it's coming from the maternal chromosome. When it comes from the maternal chromosome, Angelman syndrome is the clinical presentation. So these two disorders, it's not the deletion, it's not the mutation that determines the clinical presentation, it's where did it come from? Did it come from the mother or did it come from the father? And one thing that always helps me to remember this is the idea that P's go together. And what I mean by that is paternal deletion, Prader-Willi. So the P's go together. Paternal deletion results in Prader-Willi. That then means that maternal deletion leads to Angelman syndrome. How do we explain this? Would anyone like to explain this other than me? You wouldn't, yeah. I don't want to either, but I will, though. What we see on the normal chromosome 15 is that this region is imprinted. It is differentially imprinted male versus female. This legend down here, active versus inactive. So on the paternal chromosome 15, there is imprinting that shuts off the Angelman syndrome gene, normally. If we look at the maternal chromosome 15, we'll see that there's also imprinting, but it's the opposite in terms of imprinting. The Angelman syndrome gene is active. The Prader-Willi syndrome region genes are shut off. So everyone in this room who has normal copies of chromosome 15, what this says is that your Prader-Willi syndrome region is only expressed on your paternal chromosome 15. And the Angelman syndrome gene is only expressed on your maternal chromosome 15. So if this region is deleted, those genes are gone. So now we look over here. We have an active Angelman syndrome gene that prevents Angelman syndrome. Angelman syndrome is a result of the lack of expression of the Angelman syndrome gene. The Prader-Willi syndrome region is imprinted and shut off. It's deleted here. Therefore, the lack of expression of those Prader-Willi genes results in Prader-Willi syndrome. Then on this side, the deletion is here. It takes these genes out. Therefore, we're looking now at the paternal chromosome. The paternal chromosome shuts off the Angelman syndrome gene. It expresses the Prader-Willi, so Prader-Willi is not seen. It doesn't express the Angelman syndrome, and so we see Angelman syndrome in the resulting offspring. It doesn't matter if the offspring is male or female. In this particular case, both of these are male, but it wouldn't matter. Male versus female, they'll have Prader-Willi or Angelman syndrome. So, things that you have to look at. Where's the deletion coming from? Is it maternal or is it paternal? And then from there, put it together. You should be able to piece this together. If you say the P's go together, everything else should fall into place in terms of your understanding. So what do I mean by that? The P's go together. Okay, that means paternal deletion results in Prader-Willi. That means that the Prader-Willi syndrome genes must come from the other parent, and they're not coming from the other parent. Therefore, the maternal chromosome must imprint and shut off the Prader-Willi syndrome genes, and that's what we see. Maternal inheritance, Angelman syndrome, we look over to this chromosome, that must mean that the paternal chromosome 15 is imprinting and shutting off the Angelman syndrome genes. P's go together, and lack of expression results in the syndrome. Any question about that? There's almost always a question about that, because that's one of the most confusing genetic disorders that you can come across. Yes?
When does the imprinting occur?
Because when does the imprinting occur? And the imprinting occurs when the sperm or egg are in the process of development. So at some point, probably prior to meiosis occurring. Exactly when, I don't think we know, but at some point when that cell has committed to differentiation into a sperm or an egg. Also, imprinting is consistent too. Yes?
In these situations, it's just showing one parent is lacking the expression. Is there ever a case where both parents can lack the expression of these genes?
Is there ever a case where both parents are lacking expression? And not that I am aware of, no. Now, there are different ways. Maybe one example I can give you that's kind of similar to that is there's different ways you could present with Prader-Willi or Angelman syndrome. There's a condition that you can have which is called uniparental disomy, which is a great name because the name tells you everything. Disomy means two chromosomes. Uniparental means from one parent. So let's use that as an example. We have a child who has uniparental disomy for chromosome 15. and that uniparental disomy is maternal. Got too far away from my pointer. So, chromosome 15, uniparental disomy that is maternal. Would that result in a syndrome, and what would it be? I should have had a poll for this one. How many people say Prader-Willi? Okay. How many people say Angelman? How many people are too afraid to vote? Okay, so let's work this through. Uniparental disomy for chromosome 15 that is maternal. So those two maternal chromosomes would have the same pattern of imprinting. We would not have a paternal chromosome 15, so we wouldn't have to worry about that at all. But if we look at maternal imprinting, what is shut off? Prader-Willi syndrome region. So if you have two maternal chromosome 15s, they are both shut off for Prader-Willi syndrome region. The result then is Prader-Willi syndrome. What about uniparental disomy and its paternal? Angelman syndrome is going to result. If you can get that, and if that makes sense, and you may have to work on this a little bit. So you may have to talk through this diagram and talk through uniparental disomy, but if you can, then you'll have a pretty good understanding of it. Am I going to ask you a test question on this? Yes. Because I like it a lot. So, with this genomic imprinting, one of the important parts of this is that you can view it as a molecular signature that your parent has given to your chromosomes. There's a paternal signature. There's a maternal signature. It's thought that that imprinting is erased with subsequent generations. Somatic cells have an imprint. When then it comes into meiosis and to production of germ cells, it goes away. So a male is going to receive a maternal and a paternal imprinted chromosome 15, but a male is always going to transmit paternal signature on his chromosome 15 that he passes on to the next generation. So you, in terms of your somatic cells, the signature that your parent put on those chromosomes is maintained, but when you pass it on, you will pass on your genetic signature of being maternal versus paternal. So another way of looking at this is that here is in blue the maternal chromosome, in green, the paternal chromosome, in green the maternal chromosome. And I put an asterisk here representing a mutation. And essentially what we're saying is that as long as you're producing somatic cells in your body, that imprinting is maintained. So this mutation will always be in the context of maternal imprinting. But once you produce germ cells, and we're saying that this is a male who's producing germ cells, all of the chromosome 15s will have paternal imprinting. And now this mutation is in the context of paternal imprinting in the next generation as opposed to maternal imprinting in that generation. Can that have an effect on the particular presentation? It's possible that it could. So maternal-paternal imprinting is potentially inheritable through mitosis. But generally speaking, it is erased and then re-imprinted during meiosis. This is a figure you don't have to memorize at all, but just showing imprinted chromosomal regions that we have identified so far. And here's that chromosome 15 region. And you'll notice that there's both blue and there's gray. And essentially what we're talking about, blue versus gray, is imprinted maternal versus imprinted paternal. And so blue is maternal, gray is paternal. And so these regions oftentimes overlap, but not always. Like here's a large region where maternal imprints it one way and paternal imprints it another. But here's a region of chromosome 1 where it is strictly paternally imprinted. So these are normal processes that go on, this idea of imprinting. So exon activation is a normal process that occurs in all females who have an X and a Y, or two X chromosomes. The imprinting that we saw on chromosome 15 is a normal process. So a lot of recent work in terms of epigenetics has resulted in this big, big change is when epigenetics is altered from normal. And so that's what we're going to look at in the next few minutes, is how epigenetics can change with respect to somatic tissue, and how can that influence disease. And also the possibility that some of these differences of imprinting can actually be heritable. For a long time, it was thought that it wouldn't be, and that it would be erased at each generation, but there's more and more evidence that suggests that there is some inheritance to this. But a really good question is how far? How many generations does that go? And I'll come back to that a little bit later on. So, epigenetics, as opposed to imprinting, is this idea that we're trying to transmit additional information with respect to our genetics. More information propagated through meiosis. That is not simply the sequence, but variation in terms of the chromosomes. And some examples of this. Studies of a Dutch famine that occurred in 1944. So this was during World War II, during Nazi Germany occupation. And a number of people were placed into concentration camps, and there was tremendous famine during that time in these concentration camps. Not even just in the concentration camps, but other places as well. And so they looked at pregnancies that occurred during this time. And they could identify kind of two different populations. One was where there was a famine in the last 2 trimesters of pregnancy. So the first trimester was okay, but then famine occurred in the last 2 trimesters. And what was seen was about a 10% decrease in child's birth weight. This would be defined as SGA, small for gestational age, is what SGA stands for. So, not too surprising, the mother had famine in the last 2 months of her pregnancy. That's going to result in a small offspring. Not as much nutrient delivery to the conception. Offspring from these SGA children, in other words, essentially the 3rd generation, tended to be normal in size. Also not too surprising. Famine early in pregnancy. So the 1st trimester, but not late. Not the 2nd trimester, 3rd trimester. Normal size infants. Not too surprising because the 2nd trimester, 3rd trimester, nutrient delivery was good. So they were normal in size, but offspring of these non-SGA children exhibited a much, much higher rate of SGA, small for gestational age. There seemed to be a memory that was passed on that wasn't expressed in this generation, but reemerged in the next generation. A way to think about this is kind of this female and the conception there was exposed to famine. In some way, that exposure resulted in the transmission of genetic information to subsequent generations that essentially says, I was exposed to famine. And as a result of that, you might also be exposed to famine. And so you may want to, or it's kind of anthropomorphizing it way too much, but trying to influence perhaps patterns of gene expression so that they would be best suited for exposure to famine. Trying to set up the next generation so that it can be able to cope with the environment that the conception saw, is the idea. So early in pregnancy, normal-sized infants, but then those infants had genetic information, epigenetic information that they transmitted to the next generation. There's an Uber-Calyx study in Sweden that was done. And I'll do this rather quickly. They divided it into 3 cohorts of people. Those born in 1890, 1905, 1920. And they divided these up for very particular reasons. Is that there were differences in food availability during those times. And then they looked at access to food during what is called the slow growth period of adolescence, SGP. In girls, it's 8 to 10 years of age, and boys, it's 9 to 12 years of age. This is the particular paper in terms of the publication. So, things that they saw that were kind of interesting. When the father was exposed to famine during his slow growth period, his offspring exhibited protection against cardiovascular causes of death. So that exposure to famine that the father was exposed to resulted in epigenetic information being transmitted to the next generation, which was beneficial in terms of protecting them against cardiovascular death. Genetic information that was beneficial in terms of this. Differences in patterns of gene expression. Not as strong in terms of a p-value, but paternal grandmother exposed to famine showed a trend towards similar protection in grandchildren, suggesting it could go through, again, multiple generations. Paternal grandfather living through a famine tend to protect grandchildren from diabetes as well. Doesn't always work well. Sometimes it can be a detrimental thing, especially if the environment that the individual was exposed to is very different than the environment that the offspring is exposed to. So here, paternal grandfather had an abundance of food during their slow growth period. So they had lots of food, plenty of food. That resulted in a fourfold increase in risk for death from diabetes. So the epigenetics setting up patterns of gene expression in the offspring, which was compatible with large amounts of food, plenty of food, but that pattern of epigenetics contributing to a greater risk for diabetes. One way to explain this is through epigenetics. There could be other ways as well. So we don't know absolutely for sure, but there's a strong sense that there is epigenetic markers that are being essentially created in response to environment and then transmitted to subsequent generations. And effects on grandchildren suggest that these markers can perhaps go through multiple generations. How far? Best evidence says that about 3 generations is about it. So these are not permanent changes in terms of epigenetics that are going to be transmitted forever. 3 generations is about as far as it goes. Primary mechanisms. One is methylation of cytosines in the DNA. So the sequence is the same, but cytosine nucleotides within the DNA becoming methylated. And then histone modifications as well, post-translational modifications to histones, and then those histones being transmitted as well. Some of these modifications, acetylations, methylations, and phosphorylations on histones. There's a third mechanism involving small interfering RNA, and I've given you the figure, but I'm not going to talk about that one at all today, so we'll just leave that one alone. I'm not going to test you on that one either. So, methylation of cytosines. This is a big one. This tends to occur at CpG dinucleotides, and what we mean by that is the P essentially represents the phosphodiester backbone. So a C, if we go 5' to 3', followed by a G. And then 5' to 3' on the other strand, a C followed by a G, and that's where the methylation occurs. And so here's cytosine, and here's methyl cytosine with the methyl group added to it. Oftentimes, these methylations of CpG islands is upstream of genes within promoter regions. And that upstream of genes resulting in attenuation of gene expression. You want to associate methylation of the cytosines with decreased expression of genes. It's a silencing of the genes in that particular region. So this can have an effect on how genes are regulated. Histones. Mentioned post-translational modification of histones. Histones, hopefully you have some understanding of this already, is that they have lots of basic amino acids, a high percentage of basic amino acids positively charged, important in terms of the interaction of DNA, which is very negatively charged. And so there's a strong association between these two. What we see in these histones is that, and this is kind of hard to see in this image, but if you look, you can't really see it there, but these histones have these tails that extend beyond just the histone octamer. And these tails have even a higher percentage of positive amino acids. And it's these tails that tend to be post-translationally modified. If we look at inactive chromatin, in terms of a histone, we see that it's methylated. And just like we saw before with methylation of cytosines, methylation of amino acids in these tails is associated with inactive chromatin. And then active chromatin still has the methylations, but it has acetylations and it has phosphorylations as well. Acetylated DNA acetylated histones is very strongly associated with active DNA. So it influences how these genes are expressed. And the best understood one is acetylation of histones. So there's two major enzymes that are involved in this process. The first one is called a histone acetyltransferase, and the second one is called a histone deacetylase. And these two enzymes essentially carry out opposite reactions. So here's a lysine amino acid, maybe in the histone tail, and lysine is a positively charged basic amino acid. Histone acetyltransferase will take an acetyl group, this is your acetyl group, and it will add that acetyl group to the histone. Hopefully, easiest question of the day, what happens to the charge when you add an acetyl group? Charge goes away. Positive charge goes away when you do acetylation. Histone tails interact most tightly with DNA when they're positively charged because DNA is so negatively charged. So if you add an acetyl group, you loosen the association between the histone and the DNA. And a looser association between the histone and the DNA is correlated with a greater likelihood of gene expression for that whatever gene is near these histones. The histone deacetylase does the exact opposite. It removes acetyl groups. So if histone acetyltransferase is highly active, genes are expressed. If histone deacetylase is more active, genes tend to be shut off. And it's a modification of those histones. This is thought to change the conformation of the chromatin to a form that's more open for transcription to occur. So increased acetylation, increased gene expression. Hydrophobic hormones. I don't know if you've talked about this yet. In terms of things like estrogen, testosterone, thyroid hormone, cortisol, here is the glucocorticoid receptor. If the glucocorticoid receptor binds to cortisol, it will move into the nucleus, bind to the DNA, and associate with a histone acetyltransferase. That histone acetyltransferase will acetylate the chromatin that surrounds that region where the glucocorticoid receptor bound and allow for gene expression. Here's another one, and this one, I think, is thyroid hormone receptor. And this one is kind of interesting. Has anyone talked about thyroid hormone? Thyroid hormone receptor, probably not. Is in the absence of thyroid hormone, the receptor is bound to DNA, but it's bound to a deacetylase. The deacetylase maintains the histones in an unacetylated form, very tight association with the DNA, and that represses transcription of that gene. If thyroid hormone comes into the cell, it will bind the receptor that displaces the deacetylase, and a histone acetyltransferase binds in its place. That will then acetylate histones, and transcription will then be activated. So many of these steroid hormones or hydrophobic hormones work through their binding of either a histone acetyltransferase or a histone deacetylase. When you get into cell signaling, you'll probably talk more about that. So again, acetylation, increased gene expression. Any questions about any of that? So epigenetic errors. Got 5 minutes. Let's see how far we can get with this. Fragile X. Fragile X is a pretty common disease, and it's definitely one of those that is very well represented on the boards. Most commonly caused by a CGG trinucleotide repeat expansion. So this is a disorder that is a result of a trinucleotide repeat expansion within the FMR1 gene. So for Fragile X, the mutation is in the FMR1 gene. It is a repeat in the 5' region of that gene. Unaffected individuals who do not have Fragile X have between 6 and 50 of these repeats, of these CGG repeats. So everybody has these repeats. The question is, how many do you have? and the normal population is between 6 and 50. Greater than 200 of these repeats is strongly correlated with Fragile X in terms of numbers. If you exceed 200 and you are male, you will probably present with Fragile X. This is on the X chromosome, that's why it's called Fragile X. Greater than 200 repeats is correlated with hypermethylation at these CpG dinucleotides. So for some reason, if this repeat length in the 5' region is above 200, it becomes hypermethylated, this gene. That shuts off this gene. So then the gene goes silent on that particular chromosome. If this is in a male, he only has one X chromosome to begin with. If he has 250 repeats, the FMR1 gene is not expressed, and that results then in Fragile X syndrome. This is a Southern blot analysis, and I won't go into huge detail on this, but this is one way that you can tell that something has happened. And you see this smearing pattern. And the smearing pattern is a result of methylation of the DNA where it doesn't cut properly as a result of that. And also that we can see especially large bands that correspond to the trinucleotide repeat amplification. So Fragile X has two things essentially that happen. One is trinucleotide repeat expansion. And then the second part is epigenetic change of the FMR1 gene, methylation of the DNA and silencing of the DNA as a consequence. Okay. Epigenetics in cancer is another one that's talked about quite a bit and will be talked about more and more as time goes by. Especially with respect to DNA repair. We see quite a few mutations that occur in genes that are involved in DNA repair as it relates to cancer. Loss of DNA repair, major contributor to the development of cancer. And epigenetic changes are seen in a number of different genes that are involved with DNA repair and are thought to be a major part of cancer progression. 13% of breast cancer and 5 to 30% of ovarian cancer have hypermethylation of the BRCA1 gene. BRCA1 being a major gene that is inherited in a mutated form in hereditary breast cancer. But it's also a target in sporadic breast cancer. But it doesn't have to be mutation, it can be epigenetic change that shuts off the BRCA1 gene. BRCA1 protein, by the way, is involved in repair of DNA damage, in particular double-strand break damage. 40 to 90%, depending on study, of sporadic colorectal cancer, hypermethylation of the MGMT gene. This particular one is 0,6-methylguanine methyltransferase. This, you don't have to worry right now, at least for me, in terms of what that enzyme does, but it's involved in DNA repair. The removal of methylation on guanine nucleotides that have become hypermethylated. And so this is a major target with respect to colorectal cancer in terms of epigenetic changes. So not just mutations as a driving force for cancer, but epigenetic changes as a driving force as well. Now, this brings up an important point with respect to epigenetics, is that it's reversible. This is not changes in DNA sequence. Mutations are changes in DNA sequence. Mutations tend to be permanent within the chromatin, within the DNA. Almost impossible to change a mutation. So, can there be therapies that influence epigenetic patterns? And the answer is yes, there are. There's a drug that's called vorinostat, and the trade name for vorinostat is Zolinza. This was approved by the FDA for cutaneous T-cell lymphoma, so a particular form of leukemia/lymphoma that can occur. Vorinostat is a histone deacetylase inhibitor. So will vorinostat increase gene expression or decrease gene expression? So essentially, if we're taking that out, we are then going to trend in this direction. So acetylating, and that's correlated with looser association with the DNA, so gene expression should go up as a result of that. So vorinostat increases gene expression. And I won't go into the technical details of how it's thought to work. If you want to talk about this, we can talk about it later in terms of how this is thought to work. But again, increased acetylation, increased gene expression. This is the paper. I'm out of time, but this is the slide that I'll stop on. Is AML patients, acute myelogenous leukemia, high response rate when vorinostat is added in addition to the standard therapy as well. Okay, so I'm out of time. I will pause there. When we come back, we'll talk about our epigenome.